Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're going to continue our lesson today talking about meiosis, okay? Today we're going to talk about something called meiosis 1, and you'll know what that means in a little bit more detail as this lesson continues today. So thank you so much for making it, and let's go ahead and hop in and see what we're talking about today. So this is just a kind of overall summary of meiosis, okay? So meiosis, it kind of sounds similar to something we've talked about earlier, mitosis. Meiosis and mitosis are kind of similar. They are both types of cell division, okay? Meiosis is a little different than mitosis though. Meiosis, this is what we're using to make gametes, so sperm and egg cells. And when we're making those gametes, we want to make sure that we divide the total number of chromosomes by half. Okay, so in meiosis, we are dividing the cell, but we're cutting the number of chromosomes in half. It consists of 8 stages, okay? We're going to do PMAT twice. And so how we divide this up, we do meiosis 1, which will go through all 4 of those stages of PMAT, and then we do meiosis 2, which is the same thing, 4 more, and we do PMAT just like we did in the first stage, okay? In meiosis, DNA is not replicated between meiosis and meiosis 2, okay? So the whole reason that we're trying to cut the number of chromosomes in half is that we're trying to increase genetic variation, okay? So in meiosis, we're going to increase how different those different cells are from each other, and we're going to do this by a process known as crossing over. Now we mentioned this a little bit in the last video, I think it comes up today too, but crossing over is basically a way that chromosomes can exchange some DNA, and we can kind of shuffle the deck of genes together. This is why you and your siblings don't look just alike. Unless you're twins, you and your siblings probably look a little bit different from each other. Okay, so we're doing this through that crossing over, okay? Something else that's also going to help increase our genetic variation is something known as independent assortment. This is one of Gregor Mendel's laws, and we'll talk about that in a little bit of detail today too. Okay, so meiosis, this is the process by which we make gametes, and that's known as spermatogenesis and oogenesis. Okay, meiosis is a part of these processes. Spermatogenesis produces 4 haploid sperm cells, while oogenesis produces 1 mature ovum, which is an egg cell. Now, this can be a little confusing because even though we only make 1 egg cell through meiosis, we are ending with, 8 full cells, just 1 of them is only usable, okay? So we're starting with 1 and we're going to end with 4, okay, on both of those. Okay, so let's go through the phases of meiosis. Okay, so remember meiosis, we do meiosis 1 and we do meiosis 2. Okay, and for each of those steps we're going to do prophase, we're going to do PMAT. So we do PMAT all the way through and then we do PMAT again in meiosis 2. So here's just kind of an overview of what's happening. So in meiosis 1, you can see that we have prophase 1, metaphase 1, anaphase 1, telophase 1. So that's PMAT, okay? Meiosis 1, this is very similar to the stages that are happening in mitosis, okay? So here's what's happening in those different phases. So prophase 1, the chromosomes are going to condense and homologous chromosomes are going to pair up and crossing over occurs. Okay, so a lot of the chromosomes condensing, that happens in mitosis just like when we're doing prophase and mitosis. Some different things that are happening here is homologous chromosomes pair up and crossing over occurs. Okay, and I'll get into that in a little bit more detail as we go on. Okay, continuing to metaphase 1, those homologous chromosomes are going to pair up along the equilateral plane, okay? So another way to say that is that in metaphase, they line up in the middle. And what we really want to think here is meta, middle, they both start with the letter M, so that's an easy way to remember that, okay? In anaphase 1, remember the key word for anaphase is apart, and that's what's happening here. Those homologous chromosomes are going to be pulled to opposite poles of the cell. Okay, then in telophase 1, the chromosomes arrive at the poles and the cell undergoes cytokinesis, which is going to result in 2 haploid cells. So at the end of PMAT 1, we've already reduced the number of chromosomes in half. We just have to separate those sister chromatids apart. And that's what meiosis 2 is all about. So look, we got meiosis 2 here. We're doing those same steps. We got PMAT, but now it's prophase 2, metaphase 2, because this is the second time that we've done them. Okay, in prophase 2, the chromosomes are going to condense again in each of the haploid cells. In metaphase 2, chromosomes line up along the equilateral plane, so in the middle. Anaphase, sister chromatids are separated and pulled to opposite poles. And then lastly, telophase 2, the chromatids arrive at the poles and cytokinesis occurs, resulting in 4 haploid cells. So in meiosis, we start with one mother cell, and it's going to divide into 4 daughter cells. Those 4 daughter cells are going to be genetically different from each other. They're not identical, they're different from one another, okay? So those are the steps that are happening in meiosis as an overview. Now I wanted just to draw your attention to a couple of key words because this is going to come up later. So let's say I ask you if this thing, whatever it is this thing happening in meiosis 1 or is this thing happening in meiosis 2? Here are some key words you can lock into so that you know which phase is happening in. If you see the term homologous chromosomes, we're really dealing with homologous chromosomes up here in meiosis 1. So if I ask you something about homologous chromosomes line up in the middle or homologous chromosomes get pulled apart from one another. If you see that word homologous chromosomes, then you know we're going to be in meiosis 1, okay, because we don't have homologous chromosomes in meiosis 2. On the flip side, if you see me using words like sister chromatids, so sister chromatids line up in the middle, sister chromatids gets pulled apart to opposite poles. That term, sister chromatids, is going to let you know that it's happening in meiosis 2. So just kind of keep an eye out for those. If a question has the word homologous chromosome, it's happening up here in meiosis 1. If the question has the term sister chromatids, it's probably happening here in meiosis 2. Great, so let's kind of get into the little bit, that was just an overview. Let's kind of get into the details of what's really happening here. Okay, so remember, meiosis is just a form of cell division, okay? It's a form of cell division. It's going to result in daughter cells with half the number of chromosomes of the parent cell. And remember, we want that to happen because a sperm is going to have half the correct number of chromosomes, an egg is going to have half the correct chromosomes. When they come together, now we've got a full set. Okay, meiosis, it involves 8 stages, so we do PMAT1 and PMAT2, and it also includes 2 rounds of cell division. This is going to produce 4 genetically different haploid cells from 1 diploid cell. Let's just take a second and talk about those terms real quick. I see haploid and I see diploid cell. Okay, haploid cells are gametes. They are egg and sperm. They have half the number of chromosomes as a normal cell would. So haploid and half kind of sound alike. A diploid cell, these are somatic cells or body cells. Basically everything that's not a gamete is a somatic cell. And they are diploid, which means they have chromosome pairs. They have 23 pairs of chromosomes, which result in the full 46 that we really want. Okay, so what else is happening in meiosis? Okay, let's kind of take a second and compare this to mitosis really quickly. Okay, so I just kind of talked about what is happening in meiosis. Let's compare that to mitosis. So in mitosis, we're actually going to produce 2 genetically identical diploid cells from 1 diploid cells. It involves 4 stages and includes a single round of cell division. So here's a breakdown of the similarities and differences in between meiosis and mitosis. So in meiosis, we make 4 cells. Each cell is haploid, okay? It involves 8 stages, which are divided into meiosis 1 and meiosis 2, and then it also includes 2 rounds of cell division. When you compare that to mitosis, mitosis produces 2 diploid cells and involves 4 stages, and it only includes 1 cell division. Also, some differences here that we want to know about. In meiosis, the cells are different, and in mitosis, the cells are identical. Okay, so that's a compare and contrast of meiosis versus mitosis, and we'll compare them a lot more as the unit goes on. So let's just kind of take a look at what's happening here in these different stages. So you can see on the top row there, that's everything that's happening in meiosis 1. Okay, look at that top left picture. You see those 2 axes that are right next to each other? Those 2 axes that are right next to each other, those are known as homologous chromosomes. You'll notice that they're similar size and similar shape. Okay, one of those chromosomes comes from mom and one of those chromosomes come from dad, okay? So what we're doing in meiosis 1 is we're really dealing with those homologous chromosomes. The homologous chromosomes form, they line up in the middle, they exchange genetic material, the homologous chromosomes get pulled apart, and at the end of meiosis 1 we have 2 cells, okay? Then meiosis 2 starts. That's the bottom row. So now those 2 cells, there is no new chromosomes being made in between meiosis 1 and meiosis 2. So now those homologous chromosomes are already separated. So now what's going to happen is those chromosomes line up in the middle. You can see the sister chromatids get pulled apart. And at the end of this whole phase, we have 4 cells that are all haploid, which means they have half the number of cells as a normal body cell. Okay, so that's how we kind of break down the phases of meiosis. We say meiosis 1, which is 1 round of PMAT, and then we say meiosis 2, which is another round of PMAT. There are 8 stages total, and there are 2 rounds of cell division. Great. So let's kind of get into a little bit more detail of what's really happening here. So here's all the details on what's happening in meiosis 1. So first of all, in prophase 1, this is meiosis 1, and remember meiosis 1 is broken down into PMAT 1. So we have prophase 1. Something very important happens in prophase 1. It's called crossing over, and that's when homologous chromosomes are going to swap genetic material. So look at that picture down at the bottom. We have a red chromosome and a blue chromosome. But there in B, when they line up and form that tetrad, they're going to cross over, and now they're going to exchange some genetic material. So now the top chromosome has a little bit of blue, and the bottom chromosome has a little bit of red. So we kind of mixed up the genetic material a little bit. Okay, in metaphase 1, this is just like in mitosis. The key word you really want to know for metaphase is middle. Things are going to line up in the middle. But a really important step here is going to happen. It's called independent assortment, and I'll talk about that more on the next slide. Okay, then we'll move into anaphase. Remember, anaphase, the key word is apart, so the chromosomes are being pulled apart from one another. They move to opposite sides of the cell. And then lastly, in telophase 1, this is when the cell's going to divide into 2 daughter cells, and each with a haploid set of chromosomes. And remember, the key word for telophase is 2. So you can see the cell actually kind of pulling apart and forming 2 new cells. So let's take a second and talk about what that independent assortment is really quickly. This is kind of a breakdown of what's happening. Okay, so independent assortment, this is one of Gregor Mendel's laws, and just like the name would suggest, all this means is that chromosomes get sorted independently of each other, which means that traits are not always linked with each other. So like look at the top up there. We have these 2 different cells, and they're both heterozygous. They've got big R, little r, big Y, little y. And then the same thing on the right for that other cell. It's big R, big Y, little y. Now what independent assortment means is just because you get one recessive gene doesn't mean you're automatically going to get the other recessive gene. They are being sorted independently of each other. At the bottom, you can see that we have every combination of chromosomes that we can have. Far left at the bottom, you can see little r, little y, right? So that's 2 recessive genes, okay? But if you go to the right one, there we have big R and big Y. So we have 2 recessive alleles and then 2 dominant alleles. But then if you go over one more, that yellow third P plant, you can see that that's a recessive R and then a dominant Y, and then the last one is a capital R, a dominant R, and a lowercase y. So basically what this means is that these chromosomes can be sorted independently of each other. Just because you're yellow doesn't mean you're always going to have a smooth P pod. You can be yellow and also have the wrinkly P pod. Just because you're green doesn't mean you're automatically going to have the smooth P pod. They can also be wrinkled. So the color of the P pod does not have any bearing on whether it's wrinkled or smooth. And the same thing's happening with the green pea plants here. So independent assortment means that you can get any combination of chromosomes. Just because you're tall doesn't mean you're always going to be right-handed. Just because you have brown hair doesn't mean you're always going to have blue eyes. You can kind of get any combination of those genes, okay? And we'll talk more about this as the unit continues. So let's go ahead and let's break down each of these different stages of PMAT individually and get a better idea of what's going on here. So prophase 1, this is very similar to mitosis. These two things both happen in mitosis, okay? But they're also happening here in prophase 1. So what happens in prophase 1 that's similar to mitosis? Well, the chromosomes are going to become visible as they condense. Remember, most of the time you can't see DNA, but as it gets coiled and becomes more condensed, you can see those chromosomes a little bit better, okay? Sister chromatids are going to be joined at the centromeres, okay? Also, like mitosis, the nuclear membrane is going to disappear and the spindle fibers are going to form. So this is what's happening in prophase 1. These two things also happen in mitosis. There are other things that are happening in prophase 1 that don't happen in mitosis, and let me talk about those real quick. Okay, so these are things that are really going on that aren't exactly like how they happen in mitosis. So first of all, prophase 1 is the longest and most complex part of meiosis. It's about 90% of PMAT is in prophase 1. Okay, look at the bottom there. Something very important is happening. So when the homologous chromosomes, what you're seeing in this slide, these are homologous chromosomes, okay, which means that these chromosomes have similar size and shape, but they also have the same type of genetic material. They might have different flavors of information, but the material is pretty much the same. So this is a chromosome, this is a chromosome. When they come together like this, it's known as homologous chromosomes. Okay, and just as a quick reminder, remember that each individual side of a chromosome is known as a sister chromatid. So this is a sister chromatid, this is a sister chromatid. Together they make a chromosome. And then we have another chromosome right next to it. When these two come together, this is known as a homologous chromosome. Okay, when this happens, these two coming together is known as synapsis. And what happens here that's very important is something known as crossing over. They're going to switch that information around. Okay, so that bottom part, what is synapsis? It's when homologous chromosomes come together, they pair up, and they form a structure known as a tetrad. So this whole picture would be called a tetrad. A tetrad is made up of 2 homologous chromosomes, and each chromosome has a sister chromatid. So I know there's a lot going on there. They're kind of similar, kind of different, but we're going to practice these vocabulary terms as we go. So just hang in there with me for now. Okay, so let's kind of take another look at this. This is prophase 1 and that synapsis process. So far left over there, next to number 1, you can see that we have some chromosomes here. Okay, well, as we prepare for mitosis, we're going to duplicate those chromosomes. So now we've duplicated our chromosomes, and you can see that each chromosome has a sister chromatid next to it. Okay, here in number 3, this is synapsis. What is synapsis? Synapsis is when homologous chromosomes come together to form a tetrad. You can see here that these are kind of pairing up. We've got the pink chromosome and then the gray chromosome behind it. And same thing over here, this is another set of homologous chromosomes. These chromosomes are similar in size and shape. Okay, once that tetrad is formed, we're going to do something called crossing over. So now they're going to swap genetic material. Here in this picture, we had an entirely pink chromosome and an entirely gray chromosome. But once they cross over, they switch some of that genetic material. And now each chromosome has a little bit of gray and a little bit of pink. They've swapped that information. Okay, then we're going to start pulling these homologous chromosomes and the sister chromatids apart. We're going to pull them apart, and what we're left with is 4. These would all be gametes. Okay, they are haploid, which means they have half the number of chromosomes as they're supposed to. And you'll see that none of them are exactly alike. They're all a little different, right? Up here, the long one has a little bit of pink, and there's no other one that's like that. If you look at all 4 of these pictures, you'll notice that none of them are exactly alike. And that's what we want. We want all of our gametes to be different from one another. Again, this is why you and your siblings don't look exactly alike. Although you have the same parents and they have the same DNA to get, you don't always get the same DNA because each gamete is different from each other, okay? So this is kind of an overview of what's happening in meiosis, but I just kind of wanted to take a second and talk about that crossing over happening here in the middle, okay? So crossing over, again, this happens in prophase 1. It's a super important part of this process, okay? Crossing over can occur within the tetrad where non-sister chromatids exchange genetic material and the end of the chromatids break and reattach, leading to new genetic recombination. So again, far over here we have a fully blue chromosome and a fully purple chromosome, but when we swap that genetic material, look what we're left with. There's a little bit of purple on the blue one and there's a little bit of blue on the purple one. So that's what we want. We're shuffling the genetic material up and trying to get as much variation in between these gametes as we can. Okay, so that leads us into metaphase 1. Okay, what's happening in metaphase 1? What you really want to know is that it's lining up in the middle, right? Meta, middle, they both start with the letter M. Something else kind of important happens here, and that's known as independent assortment. Each pair of homologous chromosomes align independently of the orientation of other pairs, contributing to genetic variation. Independent assortment just means you can get any trait independently of what you've already gotten, okay? So that leads us into anaphase 1. The key word for anaphase is apart, and that's what's happening here. Homologous chromosomes are separated. They move apart and move towards opposite poles of the cell, with the tetrads being divided. In anaphase 1, the sister chromatids remain attached to each other at their centromeres. So we have separated the homologous chromosomes, but we have not separated the sister chromatids at this point, okay? This is a new term that's very important that I need to talk about. It's called non-disjunction. So non-disjunction happens during anaphase. It can happen during anaphase 1 or anaphase 2. What is non-disjunction? It is a failure to separate properly, and this results in an abnormal number of chromosomes in the gametes. So non-disjunction, it's rare, we don't want it to happen, but it can lead to some developmental issues, okay? Non-disjunction, this error is often lethal and can lead to serious genetic disorders. So let's kind of take a better look at what's happening in non-disjunction and what kind of problems it can lead to. So here's an overview of non-disjunction, okay? Non-disjunction basically means it's not separating properly, right? And so what's happening here, look, let's kind of go through this flow chart. So we've got this chromosome. It's got an X chromosome and a Y chromosome. We're going to duplicate them so that we can start to divide. Okay, so here, those chromosomes get put into the correct cells. Now what we want to do is take these chromosomes and separate them apart. The blue works just fine. Each of these gametes get one chromosome from this cell. That's exactly what we want to happen. But look what happens at the bottom. Non-disjunction means the chromosomes don't separate properly. When that happens, cells are going to get either too much or too little chromosomes. That's what's happening down here. Because these chromosomes did not separate properly, one of them got an extra chromosome and one of them didn't get any chromosomes, or got one fewer than it was supposed to, okay? This is going to cause some genetic problems down here, okay? And we'll talk about that in more detail later on. I got a whole lesson talking about what non-disjunction can lead to. But we'll kind of end real quick with this, telophase 1. This is the last part of meiosis 1, okay? Remember, we do PMAT twice. Okay, so this is telophase 1. During telophase 1, each pole of the cell now has a haploid set of chromosomes, although each chromosome still consists of 2 sister chromatids. Cytokinesis is going to occur, and that's going to result in formation of 2 haploid daughter cells. So y'all, great work. Thank y'all so much for hanging in there with me. Your teacher has some other problems for you to work on, so make sure to give that your best effort, and I hope everybody has a great day.